Okay. Okay, well, thank you for everybody that's here. We're going to wait a couple minutes to make sure that if more people are coming in, they can do so. So we'll just hang out another minute or so. And Annie, I can't see if more people are showing up now that I share my screen. So
nobody was joining for a while. We just had 3 more pop on.
So should we just get started?
Yeah, let's get started.
Okay. All right. Well, thank you, everybody. This is a seminar series that we've been doing all season long now with University of Minnesota Extension and UW Extension at UW-Madison. And we are in our 3rd or 4th webinar now, and this one is about managing grape bunch rot. We have a guest speaker today, Megan Hall from the University of Missouri. Megan is an assistant professor at the University of Missouri, and she's a viticulture researcher, so a specialist on that. And then we'll also have Annie Klodd, that you all know, with University of Minnesota Extension. And these are our 2 speakers for today. And I'll talk more about what you're going to talk about here. Let me see. Okay, so this is our agenda for today. We'll first have Annie Klodd talk to us about bunch stem necrosis. It's going to be a very quick presentation, just showing you the symptoms and telling you a little bit about that. Then we'll do a brief survey with 3 questions about the bunch stem necrosis and whether you guys have seen it, where you've seen it, and when, etc. And then we'll have the main presentation by Dr. Megan Hall that I mentioned, and she'll talk to us about harvest grape bunch rot management. And so that's really to prepare you into what's going to come at harvest with bunch rots. And then we'll follow this with a question and answer. We'll hope for about 10 minutes of doing this. As I mentioned, we have had several webinars covering all of our grapes from early season diseases, pesticide application considerations. We've talked about planting grapevines, canopy management, weed management, early season and post-bloom insects, nutrient management, and it is by no means extensive, exhaustive list of what we talked about, but just to give you a little bit of information about what we've covered. And then we have 2 YouTube channels, one, the UMN Extension Small Fruit channel, Small Farms channel, and then the Wisconsin Fruit channel, where these videos are hosted, so you can go back and look at them anytime you want. So if you do a search by those names in quotes, you'll find the channel and you'll find those videos in addition to other videos that are posted on those respective channels. And then we'll have one more webinar for grapes that will be on July 29th at 1 p.m. So we are doing these every time on a Wednesday at 1 p.m. And this one will be preparing for harvest. We'll talk about insects and also some horticulture aspects. We'll have Dominique Ebenga, that is at the University of Minnesota. She's a graduate student working on spotted wing Drosophila and Japanese beetle. So she'll probably talk about spotted wing Drosophila. I'll talk about other, so this is me, Christelle Guedot, University of Wisconsin-Madison. I'm the fruit crop entomologist and extension specialist. So I'll talk about insects that we should think about at harvest. So spotted wing, but including other insects. And then Amaya will have different topics. And we're still brainstorming on whether or not we'll have maybe other topics that we'll bring in. But Amaya will talk about possibly fruit quality or different topics that she's been thinking about. So more to come on the agenda for these. So this is what I have for now. If you have any questions, please type them into the chat. That'll be easier. If you're more comfortable with the Q&A, I'll check that too. But we'll start with our first presentation on diagnosing bunch stem necrosis by Annie Klodd. Thanks, Annie.
Thanks, Christelle. Can you see my screen?
Yes, indeed.
Oh, great. All right. So for those growers who are joining from Minnesota, you already know, you're already familiar with these symptoms because I've talked about them in a few different presentations and webinars. But for anybody who hasn't seen this before we get into Megan's presentation on bunch rot diseases, I wanted to first point out something that is not a disease. All right, so this is called bunch stem necrosis. And this is not associated with any fungal or bacterial pathogen. So it's important to know that this is one problem where if you use a fungicide on it, it's not actually going to do anything for the problem. So trying to save folks a little bit of money here, if you see this issue, know what it is so that you're not accidentally spraying something that's not going to be effective on it. I've seen people spend several $100s on Pristine to try to manage this problem when it's not controlled by Pristine or any other fungicide. However, it is kind of tricky because we don't know what's causing it. It's a physiological problem. We are seeing it more in Marquette and more in wetter vineyards or in wetter seasons, but we don't exactly know why the vines are responding to environmental stress in this way, and we don't know for sure whether it is water contributing to the problem. So a lot to learn about this issue, but what it looks like is the stems on the clusters start to shrivel up in random places. And then as a result of those stems shriveling up, the berries start to shrivel up in that order. So it's important to recognize this season if you see it, and know how to distinguish it from other bunch rot diseases. So the picture on the left, this is bunch stem necrosis. Again, the stems on the clusters are shriveling up and then the berries shrivel up because they're not getting any water from the stems. However, the picture on the right, you can see the stems on those clusters are still nice and green and plump. They have not shriveled, but the berries have shriveled up. So that tells us that this is not, the picture on the right is not bunch stem necrosis. This is a bunch rot disease. So Megan can tell you more about that. So we have a couple poll questions right now real quick because we want to, for the past couple of years, we've just been trying to get an understanding of how common this bunch stem necrosis issue is. So we have a couple poll questions, and either Christelle or I can pull those up.
If you could, Annie, that would be great.
I will.
I have them.
All right.
It's just 3 questions, really.
Yep, very brief. It should only take you a minute or two. We want to know, what state is your vineyard located in? Have you noticed bunch stem necrosis symptoms that I just described? And when did you notice them? And what varieties did you see them on?
So please notice you can scroll down once you answer the first question. All the questions are on the same panel, just in case.
While you do that, I'm going to exit my screen and Megan can share her screen.
So we will hold off on the questions until the end. That will be a lot easier, as you might get some answers during Megan's presentation. So we'll hold off all the questions for the Q&A at the end, just so people know.
Annie, I typed you a question. That's probably my fault. But I have a question about BSN. Oh, you're muted.
How do you tell the difference between bunch stem necrosis and phomopsis? I did have more slides on this that I didn't include because I wanted it to be really brief. But phomopsis, I think we usually see the symptoms show up a little earlier. Bunch stem necrosis shows up typically right before people are ready to harvest, within a week of when the berries are ripe. And not before that. So you're not going to be seeing it on grapes that are right at veraison or earlier in the ripening process. And also it happens really, really quickly. Like you won't see any issue at all. And then all of a sudden the clusters just kind of plummet.
Okay, gotcha.
Whereas with phomopsis, I think it would be more gradual.
Gotcha. Because I was looking at those pictures going, wow, that looks a lot like phomopsis. And
I know.
hate for people not to treat their phomopsis thinking that it was BSN.
Right. Yep. So thank you for asking that question. Yeah, so I'll just reiterate, phomopsis is going to happen more gradually, and you would typically see symptoms a little bit earlier.
And there's a couple comments too from both Amaya Atucha and Matthew Clark about symptoms. Phomopsis symptoms should also appear on canes and leaves. That was from Matt Clark. And then Amaya mentioned that another way of distinguishing bunch stem necrosis from diseases is that it usually appears about 2 weeks before harvest, and cluster will dehydrate very fast in a couple of days. Okay, so we're done with the poll, and so we're gonna now move into our next presentation, which is by Megan Hall, and she will talk to us about understanding and managing sour rot.
Yeah, so hi everybody, and thank you so much for having me. I really appreciate the opportunity to speak about this, and I think this seminar series that you guys are doing is great. I think it's really a fantastic way to get as many people involved and really start talking about the season and what you're encountering, and it's a great way to engage people from all over the place. So I am going to focus my talk today on sour rot because that is my specialty, but I will go in a little bit to botrytis, but mostly I am also going to talk about spotted wing a little bit because it all ties in with sour rot. So you might hear some things today that you're going to hear in the following weeks in the following seminars that you hear. But I am going to focus on sour rot because there is a lot of misunderstandings about how we manage sour rot and what it is in general. If you have other questions involving other bunch rots, I'm sure I can help answer them at the end, or Annie can definitely provide you with more information too. Okay. So I'm going to start with a brief background, talk about what is sour rot, how is it different than botrytis bunch rot, which is a big question that a lot of people have. What do fruit flies have to do with it? Which the answer is a spoiler, everything. They have everything to do with it. What are some chemical and cultural control strategies? And then I'm going to talk about wine quality a little bit, and that's brand new research that my graduate students been doing. So we're pretty excited about that. So sour rot is a major disease affecting wine grapes worldwide, close to harvest. And this comes on all of a sudden. And one of the most infuriating things about sour rot is that you may have done everything correctly all season all season. And suddenly about 2 to 4 weeks before harvest, you start to see this browning of the berries. It happens really, really quickly. Typically develops on tighter clustered varieties or injury-prone grape varieties. And so the berries push up against one another. You start to see this swelling, and then also there's an oozing that occurs as the berries sort of ooze the pulp, and that's very lovely. Also smells sour, right? The smell of acetic acid. There is typically a wound site. It can be on the outside from an insect, a yellow jacket or something like that, or a bird peck. But it also can be on the inside where you can't really see it. So it can be like a separation from the pedicel site. As I said, there is a loss of berry integrity, and that's very important. We're going to talk about that in a little bit. And there is this association with Drosophila fruit flies. And I'm going to try to convince you over the next little while that Drosophila are actually the key to this disease and key to managing this disease. So how is it different than botrytis bunch rot? So these two pictures, the one on the left and the one on the right, are what I would say as ideal pictures of both of these diseases. So botrytis bunch rot on the left and sour rot on the right. The problem is that sort of when these symptoms start to occur in both botrytis and sour rot, sometimes it's not as easy to distinguish between them. There can be just a light browning or a sort of discoloration, and that gray sporulation that's occurring in the botrytis hasn't started yet. And so it can be easily confused with sour rot. And a lot of people aren't sure whether they're dealing with botrytis or sour rot. And so they're not really sure what steps to take. So as I said, these similarities to this sour rot are really this discoloration of the berries. What happens next is where are the differences. So sour rot has a distinct acetic acid smell that emanates from the clusters. It's, sometimes many of you may have picked a botrysized berry and eaten it and realized that there's sort of like a concentration of sugars. It can taste a little sweeter sometimes, but there really isn't an off taste to it. It doesn't taste bad. Sour rot affected berries actually they have vinegar inside of them. So there is a distinct acetic acid smell and taste. As the diseases both progress, sour rot does not get fuzzy like botrytis. The only case in which this would happen is when there are secondary fungal pathogens that come and colonize those necrotic berries. So when things start to really decay and ooze and get kind of gross, sometimes you get other fungal pathogens that are just in the air, and they think, oh, hey, this is a great opportunity for me to colonize this berry because things are already breaking down. So you get things like aspergillus, penicillium, and so it'll be like black spores or blue spore, like something obvious, but it won't look like botrytis. It won't be like that nice fuzzy gray. that botrytis typically looks like. Sour rot berries also split and ooze. And so there's this loss of berry integrity that's really crucial to how we define sour rot. They also attract fruit flies. And typically with botrytis infected clusters, we don't actually see this attraction of fruit flies as much because the berries are actually staying very much intact. There isn't this splitting and oozing that's occurring on the same scale. So clusters can have both sour rot and botrytis, which obviously further complicates our issue. But in the trials that we've done, we've generally seen that whichever one gets there first is the one that the cluster is affected by. So we generally don't see a lot of botrytis and sour rot on the same cluster, but it can happen. It definitely can happen if that's, in their environment. But they are competing for the exact same space. And we've done some competition studies where we've tried to see how they interact with one another. And we've really just seen whoever gets there first is the one that gets to colonize the berries. So, there is some fine line sort of at the beginning when these symptoms are first occurring in either one of these, with either one of these diseases. But really as time goes on, and it doesn't even have to be that much time, you start to see these very stark contrasts occur. We have also tried to run experiments where we've tried to see if the color differences between a botrysized berry and a sour rot affected berry are measurable, like if we can actually take the color and measure it and see if they're different, and we haven't been able to measure a difference. So they do look very similar in that early stage. But here's how we've defined sour rot, and this is what I'm going to continue to talk about for the rest of the time here. Is that sour rot samples have to contain acetic acid, and 0.83 grams per liter sounds like a very arbitrary number. I'm going to go into a little bit how we came across that number, or came up with that number, but that's a lot of acetic acid. So we're talking about vinegar within the berries, and that's the real difference here. And then this loss of berry integrity that results in the release of liquefied pulp. The reason that we have these two different facets of this definition is that without these two, there's lots of ways that your grapes can decay, sadly. So there's lots of things out there that we know about how bunch rot, there's lots of different bunch rots we know about. There are others that we don't know anything about. But here is how sour rot is defined. And if you don't have these two components, then what you have is not sour rot. It might be something else. So how did we decide on these rules? So the 0.83 grams per liter, we came up with it because we just took a lot of samples. We took a lot of samples from 2013 through 2015 in the Finger Lakes region of New York. And we just collected everything we could, brought them back into the lab, and analyzed them for the amount of ethanol and acetic acid that they had within the berries. And the lowest amount that we had across these years while still maintaining sour rot symptoms was 0.83 grams per liter. So that's how we came up with our measurable amount and it's not that you would ever take your berries into a lab and say, well, it's 0.82, so it's not sour. This is a point that we were indicating that you had to accumulate a good amount of acetic acid in order to be considered sour rot, because actually berries accumulate sour or acetic acid very easily, and even ones that are not affected with sour rot. You can have a healthy berry that has acetic acid in it. So how did the acetic acid get there? And how we ran these experiments in which we inoculated berries with a mix of yeast and acetic acid bacteria, and then we either included or excluded Drosophila melanogaster, or your run-of-the-mill housefly, fruit fly. And then we macerated them on a daily basis to see if we could see that ethanol and acetic acid accumulate over the couple of days. So here it is over 5 days, we start to see ethanol accumulating within the grape berries. And if you look at where that blue arrow is pointing, that blue arrow is pointing to the ones where we inoculated with yeast and acetic acid bacteria, and we included flies. Whereas the green line shows the inoculation, but with no flies. You can see that green line is going way up very quickly and doesn't stop. The yellow line, however, starts to go up, but tapers off. So this kind of gave us pause until we looked at the acetic acid numbers. Well, that green arrow is pointing to that same yellow line. And basically what we see is that around day 4, the reason that the ethanol is tapering off is because it's being converted to acetic acid. So we see this conversion process occurring where the ethanol is building up within the grape berries, and then it's being converted to acetic acid. So how did the yeast and the acetic acid bacteria get there? And this has actually become a topic that I've done a lot of research on, and I'm continuing to do research on. But basically, your healthy grape berries, so the ones that are looking great, they're going to be harvested well, like everything is going well with them, they are teeming with yeast and acetic acid bacteria. They just have a lot of it inside of them. And that is what makes their healthy microbiome, just like you have a ton of yeast and bacteria inside your guts, and that's what keeps you healthy. Healthy grape berries have acetic acid and have yeast and bacteria living within them as well. The problem is when there's a wound site, that allows fruit flies and other bacteria to suddenly get in, mash up the pulp, and which is then causing this conversion from ethanol to acetic acid. So basically, we see that ethanol and acetic acid are accumulating inside these grapes, and that is what's causing this oxidation of the grape berry skin, that browning of the berry skin. But the thing is that acetic acid levels alone do not describe sour rot, and this is a figure showing all of the different types of inoculations that I conducted with different types of yeast, different types of acetic acid bacteria, and trying to cause sour rot symptoms. And what I would find was that it was really pretty easy to get acetic acid, but acetic acid levels alone did not show me sour rot symptoms. And so I came up with a disease rating scale. So we now use this in our current research as well, where 0 is a completely intact berry, and progressing through the different stages of until we get to stage 3, which is the berry has lost trigger pressure, right? So it's kind of folding in on itself, and the majority of the skin is discolored. And then into 4, where we see this liquefaction, the leaking, and then this discoloration of the berry skin. And this is what we consider, full-blown sour rot symptoms. And so to be considered sour rot, we have to see a mean disease rating of more than 3. So in other words, you need to see a loss of berry integrity in order to be considered sour rot. So let's talk about flies for a second. And so on the right here is just a sour rot infected cluster that my graduate student brought into the lab and put into a deli cup. And you can see that this is within a couple of days that we just, there are eggs and larvae and adult flies all around here. So this is how we conduct these experiments with the flies. So we surface sterilize the berries, we wound them with a toothpick, we inoculate with this yeast and acetic acid bacteria, and then we expose them to fruit flies. And really what we wanted to see in this next experiment was which life stage was actually causing sour rot. So we used adults, eggs, and larvae to do the inoculations. What we saw in all of our pathogenicity experiments that we've ever run is that we do not develop sour rot symptoms if we do not include Drosophila melanogaster. So when fruit flies are not included in the assay, we just don't get symptoms. And this is very, very important for how we're going to talk about managing this disease, is that the yeast and acetic acid bacteria alone, they don't cause this liquefaction that is characteristic of sour rot. But if you include the fruit flies, it definitely does. So we wanted to know, are the fruit flies making a microbial contribution that we're missing? Is there something here, are the fruit flies bringing something in that we've missed along the way? Is that indicating, are we missing something that we didn't originally see? And so we did something, we made azenic fruit flies. And so, and that just means that the fruit flies are devoid of gut and surface microbes. And so you can see here on the left, the 2 left-hand Petri dishes. Those are Petri dishes on which azenic fruit flies have been walking around for a couple of days. And the ones on the right are the ones where the wild type fruit flies, so the ones that are not azenic, have been walking around for a couple of days. And you can see the difference in the microbes that are growing on those Petri dishes, that they're, the azenic ones just aren't, there's no microbes in their system. And so there's nothing growing on those plates. So we make the azenic fruit flies by just collecting the eggs, washing them with bleach, and then which removes the outer chorion layer. And then we add them to the sterile media. They develop into adults. They then proceed to behave very strangely, because if somebody removed all of your gut microbes, you might also behave a little strangely. And then we add them to the assays. We also plate some out to ensure that they're clean. And what we found was that these new pathogenicity experiments where we added a combination of yeast and acetic acid bacteria with and without azenic Drosophila, we only see symptoms in the presence of the azenic Drosophila melanogaster. So even when they're missing their gut and surface microbes, they are still contributing something to the assay. So we see this acetic acid accumulate in the presence of the azenics, and then, sorry, this is what I just said, that in every permutation, we only saw the symptoms develop when the Drosophila were included. We wanted to make sure one last time that the microbial contribution of the flies wasn't laying somewhere else. And so we let the wild type flies kind of walk around on some Petri dishes for a few days. We flooded them with some water and made a little slurry. And then we used that to perform inoculations with the idea that maybe there was something we were missing there. But once again, when we added the inoculum alone, we didn't get symptoms. But when we included the azenic flies, we did get symptoms. So the microbes are not sufficient for symptom development. We need flies in order to see sour rot symptoms. So we wanted to know what life stage is causing the symptoms. And this is something my graduate student, Patrick Kenny, did in this he's finishing up now. So he did about 2 years ago. And he added different fruit fly life stages to the assay. So he added either 10 adults, 15 to 24-hour old larvae, or 15 to 20 eggs. And asked, does sour rot develop under these different life stages? And this is important for management because, well, we'll talk about that in a second. So when he took visual ratings and then through the same scale, the 0 to 4 scale, and what he saw was that the larval stages can cause disease at the same rate as the adults. And so basically we're adding eggs within 24 hours, they've developed into larvae. And then these larvae are pupating and they're living inside the grape berries. And just in that stage that they're living in the larva, they are able to cause sour rot symptoms. So in the adults, when you add the adults, they are flying around laying eggs, and then those are developing into larvae as well. And so that stage may tell us more about managing this disease. So basically, if you look at the numbers, we added 15 to 20 larvae and 15 to 20 eggs, and you see that we've recovered that many at the end of this 9-day experiment. But with those 10 adults that we originally added, we ended up with 102 fruit flies at the end of the experiment because they've gone through, there's just so many, they've laid those eggs, they've pupated, and there's so many more generations. So what is that? Like, I've told you a lot of information, so what? So first of all, it seems maybe some of you have heard some of this before, but before we started this work in 2013, we didn't have a definition of sour rot. So sour rot is characterized by this acetic acid accumulation in the berries, loss of berry integrity, and then it's caused by 4 things. It needs a wound site, yeast, acetic acid bacteria, yeast and acetic acid bacteria that you can't really control, and then fruit flies. And the flies are making a non-microbial contribution to the complex. And we now know that the larval stage of the fruit flies can cause sour rot, and this is going to affect how we manage this disease. Because if we're constantly targeting the adults, we're not actually targeting the problem, which is the larval stage. Okay, so I'm going to talk about types of Drosophila for just a second, and I know that this is probably you're going to learn more about it, but maybe it'll be good to just reinforce all this stuff in the next coming weeks when you see other seminars. So there's many different species of Drosophila out there. I'm only talking about 2 of them today. So Drosophila melanogaster are your common household fruit flies, and you see these in your kitchen and emerging from your bananas. And then there is something called a spotted wing Drosophila that I'm sure all of you are very familiar with. Melanogaster is found in vineyards and wineries. They're attracted to ethanol and acetic acid, and they feed on yeast and acetic acid bacteria. So that's their bread and butter. And their guts, our surfaces are home to many different types of yeast and acetic acid bacteria. And we've used them in most of our experiments because these are the ones we've most commonly reared from symptomatic berries in the field. But I always get a lot of questions about SWD when I give talks, because obviously it's a very popular topic. There's a lot of questions about SWD. And so just to give you a very quick overview of sort of why this, like what we've seen in regards to sour rot. So the way that it's characterized is that these the males have a large spot on their wings, thus the spotted wing. And the females have a very large ovipositor that resembles a saw. So they're able to saw into a ripening fruit to lay eggs. And they can cause a serious economic threat to many different types of fruit crops. And it can be devastating, and as I'm sure that many of you know. So both Drosophila melanogaster and D. Suzukii will feed on split, rotting, or otherwise damaged fruit. So they're very happy to see wounded fruit and split fruit, and that's going to be one of their favorite places to lay eggs, and which cause further issues. But the question of whether SWD will oviposition into healthy grapes has been really widely studied, and there have been a lot of questions and papers emerging on this topic and over the last few years. What we've learned is that there's a lot of factors that influence their preference, things like climate and environment, berry skin resistance, cultivar, ripening time, and a lot of these things go on top of one another. It's kind of compounded. So it can be that your particular clone of Riesling growing in a particular block that is prone to, you know, I don't know, you have a particular microclimate in which you're getting a lot more rain or something. Somebody, your neighbor 30 miles away who is growing that same clone may not get have as many problems with SWD as you do, or as, with other fruit flies as you do in sour rot, because they have a different environment in which they're growing that same clone. So some of the highlights from the SWD literature, and again, this is like a 30,000 foot view. There's a lot of work going on this, and not by me. So I'm mostly summarizing other people's work in this particular slide. That earlier ripening varieties are shown to be more susceptible to oviposition by SWD. So that can be problematic, especially in the Midwest and in the East, because we do grow a lot of earlier ripening varieties. And that lower penetration force, so that softer berries, and not necessarily thinner skin varieties, but softer berries are more susceptible. So penetration force and resistance are not skin thickness, but just to give you an idea of things like harder berries, Lemberger, Teraldigo, Merlot, Riesling, as opposed to softer berries, Dornfelder, Chardonnay, Pinot Noir, and Viognier. What we know is that more flies tend to emerge from injured berries than uninjured berries, right? So they, as I said before, they really prefer wounded berries. And the ability of SWD to penetrate intact berries is hyper variable, depends on a lot of factors, including the availability of other food sources. So they really like damaged fruit, we know that. But in other preference studies that have been performed, grapes don't perform very well in these preference studies. So really they prefer things like strawberries, cherries, apples, blueberries. All of those tend to be more preferred than grapes. So not necessarily their first choice to go for undamaged grapes. However, damaged grapes are very much fair game. If you're wondering what types of Drosophila species you have in your own block, almost every extension department has recommendations for different cups you can make to do your fruit fly monitoring. So this is just one illustration from Oregon State that shows homemade and different commercially available traps. You can make them at home if that's something that you're interested in. You hang them in just the fruit zone, starting a few weeks, I would say 6 weeks before harvest, and you can start collecting fruit flies weekly just to see like what you're getting, if that's something that's interesting to you. In our own experiments that we've run, these are insecticide and antimicrobial panels or treatments that we've done, we've mostly reared Drosophila melanogaster from our sour rot treatment panels, whereas the number of D. Suzukii has generally been really, really low. So even in the control over here, the control with no insecticide, we only got 5 on average so we're not really talking about many D. Suzukii. And obviously, if we applied insecticide, those numbers shrank significantly. So when we applied insecticide, we're talking about less than 2, just either type of fruit fly. Okay, so what? Again, because I'm talking a lot about a bunch of different things, but definitely preventing damage is our #1 tool for managing sour rot. And so do what you need to do in order to manage those wound sites, which I know is so much easier said than done, because sometimes you're just, especially if you're growing vignole, it's just almost impossible to manage damage, right? Because it's just those tight clusters, and we all know what happens to vignole as we get towards harvest. But things like bird netting can be really useful if you know that you have a cultivar growing in a block that's really prone to bird damage. You know, bird damage can lead to sour rot. So if you have tools like that at your disposal, those can be really helpful. SWD don't seem to prefer grapes over other fruit if given a preference. The susceptibility of various cultivars to damage depends on a lot of different factors. And in a lot of places, D. melanogaster is still the main culprit for sour rot because of they just have larger numbers, and they're just first on the scene sometimes. And so monitoring your own vineyard, your individual blocks, that's going to be not only is it a best way to tell the composition of your Drosophila community, but if you're looking for warning signs that sour rot may be coming your way, like if you're wondering whether you're going to get in a particular block, knowing that you have fruit flies in your block is going to be your best warning sign. So if you put out a cup to start monitoring your populations, the moment you start seeing more than 10 flies collecting in your cup, in the fruit zone, that's a very good indication that fruit flies are in the area. And if that block is prone to sour rot, you may want to consider using an insecticide, which I'm going to talk about next. So here's management. So one thing I asked my graduate student to do, because I just wanted to know what numbers we were dealing with. So I wanted to know how many larvae are actually in symptomatic fruit. And so he went out and he collected a bunch of random sour rot affected clusters varying disease severity, and knocked all the adults out of them and then put them in a sample cup. And at 1 week post-harvest, he rated sour rot severity and the anesthetized flies. He counted them and then removed those and then let them sit for another week. So what he saw was that even after knocking all of them out of the clusters and then putting them in a sample cup, 1 week in, he had 38 adults come out of that cluster. And those 38 were sitting in there laying eggs, which then pupate. And so when he knocked out those 38 adults, the second week, he had another 61 emerge. So, you can see that these populations, they can grow even after you remove that first population of adults. And the sour rot severity on these clusters continued to progress after 2 weeks as well. So even though he knocked out those first adults, at the end of the second week, sour rot severity had gone up. And at the end of week 2, again, after knocking out those adults, sour rot severity had increased again. So what we know is that we can, even if we remove the adults, we can still see flies emerging, and we can still see sour rot severity increasing. This was meant to mimic a little bit the effect of spraying. So if we're spraying for adults and then we wait a week, doesn't mean that we get to stop doing what we're doing. Removing adults does not stop the spread of the disease, and also does not stop this next generation from emerging. And that's pretty important for how we manage it. So I'm going to take you through quickly our disease control trials from 2013 through 2016. That's starting to sound kind of old now, but they're still very, very relevant and still the best way that we have for controlling this disease. And so what we used was several different antimicrobial sprays, and then we used either with or without insecticides. And we applied these either at 15 Brix or at the onset of symptoms. And what we saw was that every time we only sprayed an antimicrobial alone, with no insecticide, we didn't really see any good control of the disease. And you can see this with these clear bars. Those are no insecticide. And none of them are significantly different than the control. And so just putting on these antimicrobials is really not having an effect. And hopefully you're with me and you understand that by now it's because it's not controlling the flies. And the flies are the real culprits in driving the development of sour rot. Whereas, the antimicrobial plus insecticide sprays that were applied before symptoms appeared, they reduced sour rot severity by about 15% over the control. And so the one I have highlighted here is Oxidate, and that was with Mustang Max, and we had severity below 5%. And so those were our best performers. What we saw is that there was an effective insecticide in every year of our study. And there was a synergistic effect of the insecticide and antimicrobial in 2 of the 3 years of our study. But the antimicrobial alone, we only got an effect of the first year we did it. So if you start spraying, oh, sorry, just to highlight very quickly. So if you're going to choose only one, if you're going to choose insecticides or an antimicrobial to spray for whatever reason, definitely go with the insecticide because the antimicrobial isn't doing anything on its own. So if you're one of those people who decide to start spraying after symptoms appear, and there's a lot of people out there who choose to go this route because you don't want to be applying expensive materials close to harvest if you don't have to, we wanted to see what the effective different spray timings were if we were starting after we started to see symptoms. And hopefully you can see by now that when my grad student brought those affected clusters into the lab and then removed the adults, we still saw the rise of sour rot and the number of flies. And so this sort of goes hand in hand with that we just wanted to see what's happening when we're treating with insecticide after symptoms appeared. So we partnered with 2 commercial vineyards here in Missouri. We applied Oxidate and Mustang Max, and we compared their weekly sprays, so the 4 to 5 sprays, which is what we typically recommend starting before symptoms, but these people tend to spray at symptom development. We compared that to 2 and 3 sprays at different Brix levels, so 15 and 20 Brix or 15, 18, and 21 Brix. And what we saw was that there was no significant difference between 2 sprays and weekly sprays. So if you're waiting to spray until after, there's also, we repeated this in another year and found the exact same thing. So there's no significant difference if you're waiting to apply the sprays until you see symptoms develop. So if you start spraying after the symptoms already appear, more sprays do not equal better control for you. So, I would think carefully about, some people see symptoms and then they kind of panic and they start to spray a lot in the hopes that it will control their populations. But what we're finding is that more sprays they don't offer more control. That's not how this is working. So you're better off going with 2 sprays and saving yourself the material and the time, all the money, rather than continuing to spray weekly. I have to note this quickly because it's important that there's been a report of resistant D. melanogaster in New York in 2018. And I'm sure this is something you've heard of, but I like to say it over and over again because it's important. That this vineyard had sour rot and has sour rot every year quite badly. And so they were treating with Mustang Max every year, and after only a few years of using Mustang Max to treat their sour rot issues, they developed insecticide resistance. And the field flies that were resistant, they were resistant to the active ingredients in Mustang Max, Assail, and malathion. And so really only Delegate remains for this one vineyard. And we'll hope that they don't develop resistance to Delegate because then they have no tools left in their toolbox. And so what I'm trying to convey in this whole section is that the chemical control of the flies is effective before symptoms develop. That's where you have the real potential for control. But if you're spraying, after symptoms already develop, more spraying does not equal better control, which does tie in very closely with this resistance question. Obviously, I don't want anybody to just be spraying without thinking, just going forward and spraying a lot of insecticides, because there is this risk of resistance and it's serious. But if you have, waited to spray until symptoms develop, don't just spray more. You might end up with a resistant Drosophila population, and that could lead, you could lead to having no tools in your toolbox. So please remember to rotate your modes of action and avoid overspraying if you possibly can. So just to end, I have just a couple quick slides on wine quality, and then I will have time for questions. So This is again the work of my student Patrick, who did some really disgusting winemaking with sour rot affected fruit this year. So on the right-hand side, you can kind of see a healthy, beautiful vinule cluster, and then one that has sour rot. And we wanted to know how the addition of sour rot to clean grapes, to clean wine, changed the wine quality. And can we use any are there any finding agents at our disposal that might improve wine quality in sour rot affected wine? And we mostly wanted to do this because it hadn't, we were getting a lot of questions about how sour rot affects wine quality, and we can all imagine how it affects wine quality, but without continuing to speculate, we wanted to be able to put some real data behind it. So what Patrick did is that he actually made just the sour rot grapes into wine themselves, and then he sort of diluted it into clean wine. So he had a clean wine that was 0% sour rot, a low amount of sour rot affected wine, which was 15%, and then a 30% and a 45%. And these were all done in vinules. So it's not great news, I'm going to be honest. So acetic acid levels in the sour rot additions before and after fermentation. And so what we see is that in the clean wines, obviously we don't have any detectable acetic acid, yay. In that low level, we're close to 1. Medium, we're almost at 2. And high, we're almost at 2.5. And after fermentation, those numbers don't change. So fermentation didn't affect those acetic acid levels at all. What we're interested in doing perhaps in other years is doing work with acetic acid tolerant yeast to see if we could maybe reduce that a little bit. But in this first year of the study, we just wanted to see what happened and how much acetic acid we actually had in these sour rot affected wines. This low 15% sour rot addition had high acetic acid concentration. After fermentation, it was close to 1.1 gram per liter. And that's the legal limit for acetic acid. Whereas, you go to the medium and high levels and it just gets higher and higher and not so good. When we're looking at finding agents, we tried activated carbon, PVPP, and hyperoxygenation. And those were our levels before, our acetic acid levels before fermentation. After fermentation, they were no different than the control as well. And we're up around almost 2 grams per liter. So as you probably suspected, sour rot is impacting your wine quality. And the bad news is that you can probably tolerate even less than you thought you could. So our 15% was already close to the legal limit of acetic acid. And we were hoping or thinking that we could go higher than that without it impacting wine quality quite so much. But yeah, we weren't expecting that 15% would already put you over the edge. But we are still working on mitigation strategies, and so far we don't have anything hopeful to show, but we still have ideas. Blending is still obviously an option, but there is a risk of blending away your acetic acid ridden wine, and so try to be cautious with that and be mindful of the fact that there may be some funky stuff that comes along with high acetic acid levels in your finished wines. Okay, I can stop talking and hopefully have time for your questions. So I just want to extend a thank you so much to Annie for the invitation to speak, and obviously to UMN and to Wisconsin as well, and to all my funding sources, Missouri Grape and Wine Growers, Missouri Wine and Grape Board, and then my lab group, Dr. Misha Kwasniewski, who runs the enology, or who did run the enology lab at Mizzou here. He has since left me and gone to Penn State, but that's okay. And then everybody else who really contributed to this work. So thank you. Thanks very much for everybody's time.
Thank you very much, Megan. This was great, very informative, and I'm sure everybody enjoyed your presentation. We have a couple questions in the chat, and I'll have to go check on the Q&A, but a lot of the questions right now are coming with the fruit flies, obviously. So the first questions, and anybody can take those questions, obviously. But when do fruit flies first appear? How early do you need to worry about them and spray for them? You mentioned many times a Brix of 15. Is that the start that you would recommend, or earlier?
So we have generally operated around 15 bricks is when we start to see symptoms. And that's when I was doing this research in New York, 15 bricks was a pretty good bet. When I moved to Missouri, 15 bricks was a little late. So we started seeing symptoms closer to 13. A lot of that is just climate, rainfall. There's a lot of factors that contribute to that. I maintain that the best way to know that sour rot is coming is by trapping fruit flies. So just putting a few, and these don't have to be fancy anything. You put some deli cups together, smash up some raspberries, put some holes in the side, and they'll come in. And it just gives you an idea of what your population load is at a particular time. And so if you can, start putting them out a couple weeks after veraison, and just once a week, it doesn't have to be a million of them. It can be 10 of them, especially in your problematic blocks. And you're just monitoring. Hey, do I have one or none or 5 or whatever? When I've done any sort of trapping for Drosophila, you'll have a week with 10 and the next week will be like 120. And you're like, okay, something bad is happening. So you can see that kind of really steep curve happen very quickly. The problem, I'm collaborating with a lot of different researchers from across the country because we don't have a single way of recommending how to control this in all of the different states that are affected by this. How we are affected in Missouri is not how people are affected in Pennsylvania or in Virginia or in Washington. And so and then even within that, how one person's vineyard is affected is so different than another person's down the road. And so it very much is, you have to know your blocks, you have to know your vineyard, and you have to know your susceptible cultivars. And so many people only know these things because they've been burned before, right? You've seen the sour rot develop in one block and it rips through and it's devastating. Well, that's your block to monitor, right? And so the week or two after veraison, pull out your fruit fly traps, put them out, and start just peeking every few days and start seeing what you get. At the first site of, a substantial population building up, I would think about applying an insecticide spray and knocking down those populations so that you don't get to that next stage. Because hopefully I've convinced you, once you get to that level of having a substantial population and you start seeing symptoms, it's almost impossible to control. You can't go back from that. You can keep it at bay, but you can't go backwards. And so, yeah, I tend to recommend 12 to 13 bricks here as a good starting point for spraying. But I really think that scouting is the absolute most necessary tool that we have. I feel like there were a lot of parts to that question. Maybe I missed one of them.
I think I would like to say something about that because we do work on spotted wing Drosophila.
Oh, yeah.
And we've done, monitoring and all of that. And so one of the questions was about when do fruit flies appear?
Yeah.
And so what I would like to say is that if you're looking at spotted wing Drosophila and every state has been monitoring, we kind of know when they show up, and you would start seeing them in early July and then they build up. And so from mid-July to mid-September, you're at the peak. So it's a very long peak with very high numbers. With melanogaster, it's the fly you have all year long, right? So my worry about monitoring is that you will always get flies. Drosophila are going to be ubiquitous, so you're going to always have some. So that's something too that I think people need to be aware of, and to not panic too fast and spray an insecticide, because I don't know if you agree with that, Megan, and maybe we can talk about that a little bit.
Yeah.
But the flies are present. Like we can go into a vineyard where we have no problem on the grapes from spotted wing, and yet there's like hundreds and hundreds of Suzuki eye flying around and melanogaster. So that's also something to keep in mind, that you will have flies. They're there, especially melanogaster, right?
So I think that part of it is establishing a baseline for your own vineyard.
Yes.
And so, part of what I was trying to convey with sort of seeing that steep increase is that you're going to have a certain amount that lives there. And, especially if you're near a treed area where there are lots of places for them to overwinter in the leaf litter, or there's lots of, they like stagnant air too, because they don't like being blown around a whole bunch. So if there's lots of places for them to hide, there's going to always be a population. But knowing what your normal is something no one can tell you, right? So I've been to vineyards that, you catch one until you catch 100 one week. Yeah. And I've been to others where, a stable population of 25 to 50 is totally normal. But then when things like you start to see sour, it goes up to 200, and I think so knowing your normal is really important. And you can do all of that through really basic trapping, like cheap, basic trapping.
Yeah, I agree with that. I think that monitoring in your own place to know what's the kind of baseline population that you have. Mm-hmm. And I kind of like the idea of when you have symptoms, if you spray, you can do that. You can manage when you have the symptoms. You should also start maybe before, but I think that's very interesting what you're talking about. One question was, is there research showing the effect of different trellising, canopy management, and site-based quality, like sustained wind levels, on sour rot infection rates?
Yeah, I wish I'd put these slides in. So we did trials in 2015, let me think about years, 14, 15, 16 in New York. We worked with a commercial grower who had a perfect plot for doing this experiment, where he had 2 side-by-side blocks, both vignole, one trained to single high wire cordon, and then the other trained to VSP. And we monitored sour rot incidence and severity in his vineyard over the course of 3 years, and found consistently that there was more in the high wire. There was always more in the high wire. And part of that we tried to characterize the differences, to the best we could environmentally. But we're dealing, I think, with it's a microclimate issue. But I think with the high wire, because it was forming an umbrella, the air is pretty stagnant. It gets kind of warm and nice in there. It was always, if you stuck your head under the canopy, it was teeming with fruit flies. Whereas the VSP does tend to have more air movement. And so anything you're doing to encourage air movement, I think will help lower those or deter those fruit fly populations. It's not to say that that's not your only management tool, but in general, trellis systems that encourage airflow are probably going to have less sour rot. And
we'll have less flies, totally.
Yeah, fewer flies, and then as a result of that, less sour rot.
Because they like shade, they like humid environment, hot, not too hot, but still pretty warm environment. So all of that, if you have more airflow, like you're saying, it's definitely going to help with the flies.
Yeah, and high wire is actually perfect. It's lovely for them in there. They're protected, and it's warm and happy, and it's a great place to make more flies.
Another question is, how much of an effect does rain during ripening have on the spread of sour rot?
So we've been trying over the last few years to actually get, can we build a model, right? That's what I would love. I'd love to be able to say, hey, we've crossed over this much rainfall, aligned with Brix level or temperature or something, build some type of model where we can say, hey, we've had this much rain, we're definitely probably going to start seeing sour rot this week. We haven't managed to build anything concrete yet, but anecdotally, I generally see sour rot develop after large rain events close to harvest. So if you are within 4 weeks of harvest, your clusters are nearing 12 to 13 Brix or something like that, and you have a large rain event, if your clusters tend to be susceptible, if you're growing a susceptible cultivar, I would definitely consider an insecticide spray, or if you're trapping already, look for those populations to start growing, because those tend to go hand in hand. Unfortunately, though, I wish I had a model. It would be so nice to be able to point to something and show people how to do that. But yeah, I would say, yes, rain does affect sour rot. We just don't know how yet.
Yeah, and again, like you're saying, it increases the populations of flies after rain events because the humidity is really high. Yep. And they do like that. So we know that happens too.
Yep.
Another question is, good disease and insect control prior to veraison should reduce potential of sour rot later in the season? That's a question, but.
Oh yeah. So definitely, I would say that we've talked a lot about wound sites today. I would say anything that you can do to prevent wound sites is what you should be doing. It would lead to less sour rot. So, and that includes good disease management and insect management prior to harvest, right? Or prior to veraison. So, if you're not controlling your botrytis or powdery mildew or, any Japanese beetle, I don't know, anything that you have in your vineyard that could be feeding on your fruit and causing wound sites in any sort of way, if you don't treat those infections, when you get to veraison and past that point, those are beautiful wound sites for other things to come in and infect your clusters. That goes for anything. You can get secondary infections from a whole bunch of different fungi, but I would say in general, if you already have wounded berries, it's not going to be great. So yes, good management is always number one. You should always be doing all the things to make sure that you're giving your fruit the best chance.
Okay, it's 2:00, so if people want to leave, feel free to leave now, but we have a couple more questions. One is, do I understand correctly that sprays of calcium nitrate designed to toughen the grape skin don't really help?
That is up for a lot of debate. There has been anecdotal evidence that people, and I'm not going to go against anybody who's seen something work in their own vineyard, right? If you've seen it work for you and it works for you, great. But generally, no, we don't see a lot of success from toughening up the berry skins. But I don't know. I've just started to hear some things about it, but nothing that I would consider concrete now about anything that I could recommend.
Okay. You must have all seen now that there is a poll for an exit evaluation. So please feel free to answer that. Another question, and I can maybe help a little bit with that, but will sprays for other insects, wasps and multicolored Asian lady beetle, also work on fruit flies? Should these sprays be coordinated?
Yeah, you're welcome to take it as well. I would say, well, I'll defer to you. You're the entomologist.
And that's fine. I just figured I could help with that.
Great. Yeah, that's great.
It's tricky depending on which insecticide, because when you talk about any of those insects, what you're really looking at are short pre-harvest intervals, because we're at harvest time. And so, for example, for multicolored Asian lady beetle, a lot of them are neonicotinoids, which do not work well at all on fruit flies. On wasps, it's another question too, because there's no insecticides that are directly labeled for wasps. So there's no label that would have wasps under them for grapes. So you kind of have to go with the insecticide that either have those different insects on the label already there. But if you have, the main point would be that if you're looking at a neonicotinoid for treatment of multicolored Asian lady beetle, that will not work for your fruit flies. If you're talking about pyrethroid, for example, or if you're talking about like Mustang Max, for example, more of those broad spectrum, yes, they're likely to cover all of your insects at the same time because they work on their broad spectrum. So they work on most of your insects. So it's something that overall you have options that will cover all of your insects, but neonicotinoid is one, for example, that I wouldn't apply thinking you're going to control your fruit flies because they don't work.
There was one question that came up at the beginning that I think we might have missed, and it was about bunch stem necrosis.
Yep, right, indeed.
Is it suggested to harvest immediately when there is bunch stem necrosis noticed? Hopefully the person who asked this is still online. So since bunch stem necrosis is such a new problem, and by that I just mean it's only the past few years that people have started noticing it really bad. There hasn't been time for much research to be done on this problem, and we have to be able to get the research done in order to provide more official recommendations. So I'll just preface it with that, we don't have official recommendations for this disease yet because we haven't had time to research it yet. But I would say that it would be a good idea to harvest immediately after you start seeing bunch stem necrosis, as long as you can really confirm that that's what it is. And if you need to send a photo to one of us, you can do that, to just have us say, yep, that looks like BSN, to make sure that that is what it is before you go ahead and do an earlier harvest. Once you start seeing bunch stem necrosis, it typically just gets worse and worse. I mean, the clusters that are affected are not going to become unaffected, of course, and it typically just kind of spreads. People will say within the course of 3 days, it went from nothing to 50% of the grapes affected. The amount affected can really vary, but it's not going to get better. And so if you do decide to harvest immediately, once you start seeing a bunch of those symptoms, you might need to add more sugar in when you make the wine because maybe your bricks aren't correct yet. You might need to correct pH somehow. I'm not a winemaker. That's not my department, but you might just need to fix that in the winemaking process or use those grapes for something like a sparkling.
There's another question actually that I had missed. Is there any evidence that bunch stem necrosis could be a variant of trunk disease?
Oh, yeah, we don't have any evidence of that at this point in time.
Oh, and Amaya wanted to make some comments as well. So Amaya, feel free to unmute yourself.
Yeah, I want to make actually some comment about the trunk diseases. What we've seen here in Wisconsin on some of the Marquette blocks that we had is that some of the vines that had a lot of trunk diseases, when we remove some of the cordons and we retrained and form new cordons, we saw substantial decrease of the stem bunch necrosis that we used to have a lot in Marquette every year. So I don't have, direct evidence. I haven't done the research, but what I've seen is that vines that have some trunk diseases might be more susceptible, and by renewing cordons, we, at least here in our research blocks, we were able to overcome some of that damage.
I would say too that there's a lot of nuance to that because all of the vineyards that we sampled in our trunk disease survey had some grapevine trunk disease. So there's probably a lot of different factors that could go into that. If there is an association there, there's probably a lot of factors influencing that. Because we found trunk disease at every vineyard we looked at, and only a portion of those vineyards had bunch stem necrosis.
Thank you, ladies. Okay, last question. As a follow-up to the rain question asked earlier, do you notice an increased level of sour rot when you have a very wet vineyard season overall, not just at harvest?
No. I would actually say no. So we've actually, that's kind of an annoying thing, is that sometimes we have, okay, so for instance, last year in Missouri, we had record flooding. I mean, we had terrible, terrible flooding and so much rain from the early part of last year all the way through the summer. And then it just kind of stopped raining. Like, not entirely, but we continued to get rain, but it wasn't terrible, not like it had been. And we had very low sour rot. We had almost no, like, sour rot was negligible. So it was kind of annoying to feel like, well, here's something that we're almost sure is, directly affects this, and it didn't really. So I think it's not the only factor that we can look at. And overall wet years don't mean that we're going to have more sour rot. It's more of, I think it has to do with more the specific timing of those rain events closer to harvest, probably partnered with humidity and temperature. wind speed. Like there's probably a whole bunch of different components we haven't even factored into our model yet that would inform that. But no, generally no.
Okay, well, I guess this will conclude our webinar today. Please remember that our next webinar is July 29th. Is that correct, Annie? I was trying to find that, but yeah, okay. So July 29th, I would like to thank Megan very much so for joining us and providing a lot of great information on sour rot, and Annie about bunch stem necrosis. And the webinar, as we said, will be, the recording will be posted on the YouTube channel. So please feel free to go back there and get more information that you might have missed. And thank you everybody for participating in this webinar.
Thank you.
Thank you.
Thank you. Bye-bye.